Well, thank you to everybody for coming today to this session. We have 3 excellent speakers for you today. We start with, I apologise for the, I meant to check with you on pronunciation first, but we have Ellerin Iche? Eha. Apologies. Aiha. Okay, there you go. Apologies. Who's from the Estonian Medicines Agency, and she's going to give a really excellent overview of the EMA draft quality document that she's been leading. And then we have Fredrik, who many of you will know, who is from 5QBD Biotech, and she's going to go through the alignment of the EMA quality draft with sort of innovator expectations and where the gaps are. And then finally, Professor Laurent, thank you, is going to talk about the pivotal role that phage susceptibility testing plays in this area, and some of the challenges and limitations of how they are being addressed. And because I know that regulation sometimes can seem a daunting subject, we wanted to remind you that regulators are humans too. We're real people. And so what I'm going to do is, before each speaker, I will introduce them, and I'm going to tell you 3 things about these people. One of these things is a lie. At the end of their presentation, we'll vote to see if you can work out which one it is. So, first, we have Ellerin, who is, she's a quality assessor for the biologics at the Estonian Medicines Agency, where she evaluates quality documentation for marketing authorization applications. Her speciality is monoclonal antibodies and recombinant proteins. She's a member of the EMA Biologics Working Party, where she leads on quality guideline drafting for PTPs. She did her PhD in cell biology and MSc in biopharmaceutical science. And here are your 3 facts. She likes badminton, with a strong preference for winning. She dates somebody who plays bass in a rock and roll band. And she runs a YouTube channel debunking scientific health myths. So those are your 3 facts. Please.
Hello, dear audience, and thank you, Carmen, for a very kind and fun introduction. I'm very honored to present the draft guideline on quality aspects of phage therapy medicinal products. We have been working on this guideline with EMA and EU regulators for around 2 years now, and I'm happy that we now have a major draft ready. So bacteriophages are viruses that exclusively infect bacterial cells, replicate within them, and often cause the killing of their targets by lysis. And often they have very narrow strain-specific ranges, which can be advantages, but also introduces some challenges for their development as products. Phage therapy has been known already more than 100 years, but its use was largely abandoned after the advent of antibiotics. However, in the recent years, the interest in phage therapy has resurged once again. And as a regulator, and regulators in general, we are interested in how to ensure the consistent quality, safety, and efficacy of phage therapy medicinal products. Phage therapy has 2 approaches. We have standardized phage therapy, which where the product has a fixed qualitative and quantitative composition. They are ready to use off-the-shelf products, which fit the current marketing authorization framework. They follow the conventional drug development pathway. And the standardized phage therapy is more suitable for the development of phages which have broad host range, so they can be used for a larger patient population. Individualized phage therapy, on the other hand, is tailored to individual patients. In this approach, the phages for treatment are selected on the basis of testing the bacterial isolate from a patient against a larger phage collection. And the product is formulated for each individual patient separately. So while the standardized phage therapy fits the current regulatory framework, it is much more challenging for the patient-specific preparations, in which mostly alternative pathways are used. So when we look at EU regulatory initiatives, what regulators have done to facilitate or support the field, we can see that on this top timeline that for a long time nothing really happened. However, since 2023, there is a good pace of acceleration. So in 2023, the EMA published a guideline on veterinary phage therapy medicinal products. In 2025, ETQM published a general chapter for phage therapy medicinal products, and also is now preparing another general chapter for phage potency determination. And those two are really the key milestones since for the first time in the EU we have basic quality standards for phages, so the quality expectations and also standardized potency assays. So the focus of my talk today is the EMA guideline on quality aspects for phages, but the EMA and EU regulatory network are also working on a horizon scanning report for phage therapy, and the aim of this project is to map the current landscape, challenges and opportunities in the field. So let's dive into the guideline now. The scope of the guideline is to clarify what EMA and regulators expect from a marketing authorization dossier for fixed composition PTMPs. And why only fixed composition? This restriction comes from the legislative barriers that I briefly come back to later. And the aim of the guideline is to clarify the current regulatory expectations. Its aim is not to introduce any new legal requirements. And the guideline is currently on public consultation until the end of April. And I use this opportunity to invite everyone involved in phage therapy to contribute and share your thoughts on the draft. So we really want it to be a collaborative effort. So the guideline is applicable to only strictly lytic phages that are mainly intended for the treatment of bacterial infections. In scope, we have natural phages, phages with synthetic genomes, chemically modified phages, and also those genetically modified phages which do not classify as gene therapy medicinal products. For those genetically modified phages, which are classified as gene therapy products, for those, the principles of the guideline generally apply. However, since those products have a different legislation, then also the ATMP guidelines and legislation has to be considered. The principles of the guideline also apply to self-reproduction systems and investigational products. However, phage-derived products such as lysins and enzymes are out of scope because for those, the guidelines for standard protein-based therapeutics can be followed. And patient-individualized and magistral preparations are also out of scope. So why do we only address strictly lytic phages? There are several reasons, and most importantly, only the strictly lytic life cycle ensures the direct killing of their target cells. And also, they are self-limiting, so their propagation depends on the presence of bacteria. And also, those phages have a lower risk of horizontal gene transfer. Temperate phages, however, are capable of lysogeny, so they can remain dormant instead of directly killing the target, and it makes their efficacy much more unpredictable. And they also come with a higher risk of horizontal gene transfer and lysogenic conversion. We put a lot of effort in the quality of the starting materials, which are normally bacterial cell banks and phage seedlots. And the guideline does not introduce any new requirements, and it is fully aligned with the ETQM general chapter. From the quality, we expect, of course, general information about the seedlots or cell banks. We expect the identity to be established based on full genome sequencing, also quality, microbial quality, and potency, and for bacterial also phage susceptibility. And then an important part is a genomic safety screen, which means full genome sequencing and its annotation and analysis. And the aim of this is to make sure that there are no unwanted sequences in the genome. For phages, the absence of lysogeny models should be verified to confirm the lytic nature. We also expect that detrimental genes are absent from the sequence. And for bacteria, the absence of prophage sequences should be shown, and also the absence of detrimental genes. However, if those detrimental genes or prophage sequences are detected, it does not necessarily block the use of those cells or seedlots. We expect those risks to be evaluated. And here I try to explain how this should be done. So, and I use an example of, it does not work, prophage sequences in the host bacteria. So let's assume that you have found an ideal production bacteria. However, there is a prophage found in the genome, and attempts to remove this prophage have been failed. So the next step would be to analyze the risks and evaluate them. And so what might happen is there might be prophage proteins or prophage DNA or induced prophages in your final finished products. So all these risks should be carefully evaluated, and then the next step is to evaluate whether those risks can be detected, whether they can be reduced or managed, and then a decision. If the risks are considered unacceptable, considering also the patient population and the intended use, then the risk assessment should be either re-evaluated or this host should be abandoned. If the risks are considered acceptable, then in the regulatory dossiers, we would expect to see a scientific justification for the use of this host, complete risk assessment data, full characterization of the prophage, and also adequate control strategy. So from the characterization, normally in the regulatory dossiers, it includes the characterization of the phage itself, the active substance, and then impurities. So what else is present? For the characterization, we expect to see information on phage identity and its structure, also taxonomic classification. From the activity in the characterization section, we are interested in having some understanding of the host range, and also if the company claims that their phages work also on biofilms, then this should be supported by data. We also already talked about the safety and genomics. So here, what is important is that the genomic characterization can be done at a master seed level, provided that the genetic stability is demonstrated. For the impurities, the product-related impurities, for example, aggregates or non-effective particles, should be characterized, and process-related impurities for those such as host cell DNA, host cell proteins, endotoxins, or residual regions. So for those, we expect to see process clearance, the removal capacity of the process. So there is no really extra regulations. We try to be reasonable, and these are actually standards like for any other biologics, but just adapted to phages. Specifications, so we give some guidance for both establishing drug substance and finished product specifications for release and also stability. And there are some minimum expectations, so the core quality attributes that should be included every time. And then for others, for example, process-related impurities or product-related variants or dosage form attributes, the decisions can be case by case. And I will give you an example, and this time using host cell protein impurities. So generally, host cell protein testing is included in the drug substance specification for biologics, and this is also mentioned in the AMR Q&A for biologics. However, in case of phages, we have to think whether the residual host cell proteins from the finished product, from the vial, is it meaningful, considering that during the lytic activity of phages, there will be a huge amounts of host cell proteins released at the infection site. So for this particular example, host cell proteins, the risk to patients depends on the administration route. So for oral administration, the risks of inducing immune responses are much lower compared to, for example, systemic administration. So in certain cases, depending on the administration route, routine host cell proteins might not be obligatory if the clearance is properly validated. So the process always has to be capable of removing the impurities, and this has to be shown. And finally, I have some considerations we have established some considerations for phage cocktails. So the phage cocktails, they can be, of course, advantageous in certain cases, but they also increase the complexity. So we expect to see rationale for combination of the phages. The number of phages should be justified because the complexity increases with each additional phage. The interactions between phages should be discussed. We are interested in knowing about the synergy, antagonism, or maybe the phages do not interact, so those should be investigated. And for the potency, so if there is a phage cocktail, then the specific potency for each phage in the product should be tested using a suitable host bacteria. So We hope that this guideline will clarify the expectations and increase transparency. However, it does come with limitations. So first, it only addresses quality expectations. However, for clinical and non-clinical areas, the guidance is still needed. And the other limitations are related to the legal framework. So first, as I mentioned previously, the marketing authorization requires defined qualitative and quantitative composition. So it's suitable for the fixed composition products, but not for patient-specific preparations. And also with regards to life cycle management. So in the case, sometimes bacterial resistance emerges, which might require phage updates or replacements post-authorization. However, those changes are resource intensive under the current rules requiring a line extension. However, recently the new pharma regulation was, the new pharma directive was adopted, and this pharma update also foresees tailored pathways for phage therapy medicinal products. However, the details of this adapted framework are not yet known. They will be implemented a bit later. And I hope that the European regulators and also other stakeholders will have an active and coordinated part of this process. So this guideline provides clarity on quality expectations now, but we need broader solutions which need policy life cycle tools and also continued evidence generation. And I would like to thank my colleagues who contributed to the development of the guideline, and also to EMA and EDUCOM colleagues. And this is all from my side, and as you can see, I like graphical art.
Thank you very much. I'm sure you've all got questions, but we're gonna get through the talks, and then we're gonna have questions at the end. And so, coming back to my facts, I gave you 3. Okay. So, is Ella in? She's a keen badminton player with a predilection for winning. Her partner plays bass in an amateur rock band, or does she runs a YouTube channel that debunks myths around misinformation in science? Which of those? Who thinks it's number 1? Is the lie? Number 1, okay, yeah. Doesn't look like a badminton player, right? Yeah, okay, fine. Number 2, married to a musician. Yeah, yep. And number 3, runs a YouTube channel debunking. Yeah, you're right, it's number 3. But I was really sad because I actually thought this was true and I got really excited, and then you were like, I'm really sorry, but I don't. I was like, oh, damn it. Okay, right, thank you so much. So our next speaker is Frédérique from 5QB Biotech. So she's a regulatory scientist with a specialization in innovative biological medicines, associated diagnostics. She has 25 years across development, manufacturing, quality systems, and regulatory affairs, very knowledgeable. She leads engagements with has led engagements with European and US regulators, and she's the co-founder and strategic lead of the phage ACT. So hopefully you've seen her emails. She's putting in quite a lot of effort to herd what I would describe as cats, trying to get everybody into the room and talk about these things. So I think you should be applauded for your efforts there. Yeah, and so your 3 facts. So at the end, I'm going to ask you. So she enjoys skiing and rock climbing. She once considered becoming a regulator. And she owns a bearded dragon called Frank. Those are your 3 facts. Come back to it. Thank you.
Good afternoon, everyone, and thank you, Carmen, for this nice introduction. So, because we all love regulatory, let's continue on this topic. I will talk about the regulatory foundations and the remaining gaps to unlock the full potential of phage therapy. I mean, to pass from few patients treated to a bigger scale, products on the shelf, which will require frequent changes. So I will come back a little on the regulatory landscape and how the unique biological characteristics are translated into these guidelines. Then we will see the remaining gaps through FEDG Act contributions, and I will briefly mention some main challenges regarding the phage therapy clinical development before the last takeaways. Okay, regulatory landscape. So, thanks to Ellerin, now the regulatory landscape holds no more secret for you. We have the same kind of slides. I prefer yours, Ellerin, definitely, nicer. So, everything started in 2023 by this amazing EMA guideline on the veterinary field. And since then, each year, a new regulatory steps. So, one concept paper, 2 general chapters, and this EMA guideline for human field, very well presented by Ellerin. I will talk briefly of all of them. What I won't cover today is these regulatory considerations for phage therapy from MHRA, but it's the right place and the right moment to ask any questions. And there is as well Matthew in the room who leads this MHRA survey. Okay, let's start. So I will start by these 2 key phage guidelines, the 2 first published, and because they can be considered as a reference for phage therapy. Why? Because they take into account the phage specificities. It's not implicit, it's written. Look at. So, EMA guideline, veterinary field, which covering quality, safety, and efficacy. From the first page, first paragraph, everything is said. We are talking about the trio, regulatory, technical, and scientific considerations, which is not so common for guideline. So that is good, and we need that for phages. Then, this dynamic interaction between bacteriophage and bacteria is mentioned, with an acknowledgement of the resistance. As a result, the product is expected to require frequent changes in composition. Don't worry, I didn't copy each text and each guideline. I just extracted for you the key wordings. From this recognition of these frequent changes in composition, we have a specific paragraph on the flexible composition, quantitative and qualitative. And when we are talking about qualitative, it includes phages not used in key safety and efficacy studies. And this is to match with epidemiological needs and bacterial disease outbreaks. In this guideline, more than that, there is this sentence: such customization of phage products doesn't require variation applications. 2 important things. First, this is possible in the veterinary field because the veterinary legislation allows it, and as you saw it, soon on the human field as well. We will have it. And another thing, it was during this meeting at the EMA in Amsterdam in 2024, when this topic was presented, the question was raised, okay, but how it will work in practice? The answer was simple: come to see us, discuss, and it will be handled case by case. And even in this guideline, at the end, there is a lot of case by case. Okay, now, just to jump to this other guideline, phage therapy medicinal product at the EDQM level, so European Pharmacopoeia. Very interesting is the general chapter. They didn't set up a monograph, so it's very good because a general chapter is not binding, legally binding. Okay, so that provides flexibility. I will not highlight what we can see in the general chapter, you know, identity, potency, impurity, etc. I will highlight 2 things. First, this characterization. As explained, detrimental factors should be avoided. unless otherwise justified and authorized. And this is very important and interesting because few years ago, prophages were not at all welcome in EMA, okay, inducing blocking issues. Now, what we have this in the guideline of EDQM, unless otherwise justified and authorized. And today, you had a good example of the risk assessment to perform. So, good advancement. Okay. Now, even in this text, we have this adapted product. So it's recognized that we need evolution of the product. And this shows that the field is maturing. So what we see regulatory openness, we see understanding of the fetch specificities. Of course, it remains some gap and remaining topics to address, and there collaborative work becomes important. Good news is what we are doing in Fetch Act. Welcome in this second part of the presentation. Okay, so Fetch Act, created in March 2024, so it's a BIM Alliance task force dedicated to fetches. Our aim is to shape the regulatory landscape and facilitate access to fetches. We addressed different topics, and the past 2 years, the focus was on regulatory. We commented each new draft of guidelines, and we have written an internal white paper regarding the platform-based approach for PTMP, Fetch Therapy Medicinal Product. So, what I will mention now is different contributions and our comments on this guideline. I will start by this text from EDQM, European Pharmacopoeia. Last year, we have contributed in July 2025. So, once again, even for the most critical potency essay, they set up a general chapter, not a monograph. That is good to give flexibility. Now, in this text is mentioned, what we want to see is replicative activity by measuring plaque formate unit. And this is important because for the PST, fetch susceptibility test, the logic could be different when we want an high throughput method and as well a quick result. But here, it's plaque formate unit. And the gold standard means often old method. It's a double layer agar plaque essay. What we asked is that this spot test, until now as an alternative method, should be positioned as a compoundial method, as a variant of the method. And that will be helpful to, and it will facilitate, its implementation, because it will avoid this overwork to validate this alternative method. And by the way, it's a plaque essay, high throughput method, used commonly when suitable in phage therapy. The second comment was this titration. We asked to not focus on the production strain because sometimes it's not suitable for this potency assay, and to rephrase it as establishment of a reference bacteria strain. Just to give you an example. Now I will take a little more time regarding this concept paper we commented in March 2024. It was issued to establish the guideline presented by Adrienne. Why I will take more time? Because the gap identified at this time still shapes the fetch development mindset. It was interesting because from there, the two approaches were mentioned, the fixed cocktail and the personalized approach. What we asked, first to extend the scope, because now the non-clinical and clinical aspects remain the major gap for phage therapy. We ask as well a clarification of the definitions. Still now, around the table, nobody has the same definition and understanding regarding personalized, individualized, and magistral preparation. Good, the third point is resolved. Now we know that a phage therapy medicinal product should be compliant to GMP and European Pharmacopoeia. So that is resolved. The two elements following are very important. for phage therapy. Why? Because we want to integrate any new phage in a streamlined manner. So for this, we need to develop this kind of concept, platform-based approach across development, manufacturing, and QC, but as well to review the regulatory process to assess and approve this kind of product. So this is the two main points, which are still open. Now, regarding this EMA draft, I will not compete with Ellerin to present it, not at all. I will neither present the Fetch Act because the discussions are ongoing. What I can say, it's a welcome guideline. We all expected it. We appreciate it, and it's open. There are a lot of unless otherwise justified and accepted. And what is very, I would say, crazy, is this last section regarding regulatory considerations. Because I explained this flexible composition is not yet in this guideline, but there is a specific paragraph at the end just to say, hey guys, we know what you expected, but we cannot provide you regarding ta. So I never see that in a guideline, so thank you. We cannot be upset. It's a pity. Okay, so good things and flexibility, openness, so it's good. Of course, there are some comments, and just spoiler, I remember this first meeting where the minutes were, okay, 2 warning points, which are transducing gap capacity regarding this kind of transduction studies. It's a critical, tricky topic. And the second one is prophages, but today some answers were given. Okay, so key messages, 3 slides. First, the clinical development overview, and after the takeaways and the things. Okay. These slides doesn't come from guideline, from my mind. A lot of information everywhere, quite ordered. I will not come back on regulatory and CMC, the advancement and the gap. Let's focus on scientific and clinical challenges. When I'm saying scientific, it could become from non-clinical and clinical studies. I would highlight 3 points. First, this immunogenicity. We need to understand more the phages, how it works, and what it can bring. Okay, so we need more data, PK/PD data, immunogenicity assessment, and I remind for the moment, in vitro and in vivo predictivity is not yet demonstrated. On the clinical challenges, I would focus on the standardization of therapeutic schemes, which is today not yet done, the dose rationale, which is very tricky to write, and it's very difficult, as you know, to demonstrate efficacy on top of the standard of care. And the last but not the least, my favorite topics, the PST, phage susceptibility test, which gathers all challenges, regulatory, technical, scientific, and clinical, and regulatory. And nothing is answered for the moment. There is no reference method, no standardized protocol, no breakpoint. But looking forward to Frederick talks in a few minutes. So we have seen regulatory advancement, openness, understanding of phage specificities. What is sure, we need to rethink the way we develop and assess, approve these kind of products. For this, we need a greater openness across the ecosystem, flexibility in development and manufacturing, thanks to platform-based approach, and to streamline this regulatory pathway to assess and approve this kind of product, but of course to standardize this PST and to be innovative in the clinical design. And to finish, I would like to thank all these Fetch Act experts. It's a real pleasure to lead this group. It's a dynamic group, open, we exchange freely, we challenge ideas, and even when we are a little disagree, we find a compromise, so thanks to all of them. Thank you.
another excellent talk from Frederic there. And I would encourage all of you to engage with PACT and to use that tool that Frederic is providing. And so finally, on to PSTs. Oh, you're right. How could I have forgotten? You must be hanging on the edge of your seat. Right, okay. Fact 1, enjoy skiing and rock climbing. Is it true? Is it false? It's true. Is it true? It's true. Okay. Once considered becoming a regulator. No, just a single person that might have actually been pointing. Finally, owns a bearded dragon. Yeah, that's true. Yeah, you're right. Well done. You guys are good. Can't get anything past you. Right. So yes, sorry. On to our final speaker, Professor Laurent. Your bio is quite long, but I'll. He's done a lot. Professor in clinical microbiology at Lyon University, Institute of Pharmaceutical Biological Science, hospital position, co-PI on Staphylococcus pathogenesis team at the Centre for Integrative Research in Infectious Diseases and Immunology. You try saying that. His speciality is epidemiology and looking at the mechanism and diagnostics of virulence with a passion for AMR and staphylococci. He coordinates phage in Lyon and where he's focused on the selection, characterization, production, manufacturing of phages for the treatment of patients with a number of MDRs. Established the first French GMP phage production platform, developing innovative phage susceptibility testing assays, and chair of the UCAS subcommittee for phage susceptibility testings. So as I said, quite extensive. So where to start? Okay, so he organised a national phage building contest out of Lego. That's the first fact. The second fact is he spent 3 days in jail. I won't say why. And the final thing is that he inherited a very rare stamp called the SS Pasteur stamp from 1941, which is worth over 20,000 euros. Please.
Thank you. Thank you so much for this introduction. Okay, we will finish this session with John Lennon. You know, of course, the iconic song of Imagine. So imagine antimicrobial chemotherapy without antimicrobial susceptibility tests. So without this diffusion, without liquid media, without e-test, and without the data from the OCAS to define susceptibility and resistance. It's exactly what we have with the phage therapy. If we have the same musical metaphor, it's like something like Sounds of Silence or Welcome in the Jungle. So is the reason why I have this title, very long title, The Pivotal Role of Phage Susceptibility Testing in the Regulated Use of Phage Products, and what still limits its implementation today. So we work in Lyon on phage therapy, and we have some experience in this field. So as you know, phage susceptibility testing is presented as a precision medicine. That means it makes sense with the scientific accuracy of treatment. We try to use a highly specific phage to target one or few bacterial strains in patients. It is also presented as a personalized therapy. It's more focused on patient-centered approach, and we try to give into account all the data we have for the patient to select the best phage treatment in terms of safety, efficiency, and acceptable for what is acceptable for the patient. So it's the reason why we need to have susceptibility to be able to do personalized personalized treatment and precision medicine. The problem with phage is for phage activity. Phage activity depends on the recognition of the bacterial receptor by the phages, and sometimes there is some mechanism of resistance of this recognition due to the point mutation of the receptors, or a way to mask to mask the target of the receptor for the phages, or the receptor is no more expressed by the bacteria. But also we have expression of natural or acquired anti-phage defense system, and there is many different systems, and many are uncharacterized, and we have each week we have new mechanisms described in the literature. We have also, of course, the CRISPR-Cas system. We have also the abortive infection. The bacteria is able to suicide before to expand the number of phages to protect the community, bacterial community, and so on. So many different mechanisms of resistance, and it's the reason why we need to have phage susceptibility testing to be able to adapt the treatment to this kind of mechanism present in bacteria. So, and this kind of mechanism of resistance defines the phage spectrum. And for Staphylococcus aureus, for example, in our lab, we are able to select only 4 phages that are able to cover 91% of the Staphylococcus aureus diversity. And because in part because the receptors for Staph aureus for phage against Staph aureus are very limited in terms of diversity. Most of the receptors are on the wall teichoic acids. But it's not the same for Pseudomonas aeruginosa. It's our experience. With 4 phages, we are able to cover 74% of the diversity of our large collection of isolates. But if we want to reach 92%, we have to add 7 phages, and we gain only 18% if we added 7 phages. So after 4-6 phages, to gain something, you need to have many phages. And it's worse with E. coli, for example. Here you have a table. If it's green, that means that the efficacy of the phage is very good. In blue, it's middle, and red, the efficacy is not good. And you could see here that you need to have 5 phages to cover 64% of the diversity of the E. coli. And as you could see here, it depends of the activity depends of the genetic background. And you could see here when you have a phage active against one strain of this background, ST38, the second strain is not covered. That means the activity is not sequence type dependent, so it's very difficult to cover all the diversity of E. coli. And it's really an issue. And without phage susceptibility testing, there is a huge impact on the design of phage therapy. Because of the limited coverage of species diversity by each phages, you need to combine phages, active phages, against a maximum number of isolates. And so you could use only a fixed cocktail with many phages and too many phages. And the problem is when you put too many phages in the same time, you have some trouble with instability, because phages are positive charge and negative charge, and they try to aggregate all together. And when you mix different phages, it's more the case than when you have just one kind of phages in the same vials. The second thing, it's also that if you use a combination of phages or many phages, you will see an immunization of your patient. So, and the antibodies will neutralize the effect of phages. It's not an issue for acute infection, but if you have to treat chronic infection like cystic fibrosis, it could be an issue. So the fact not to have a phage susceptibility testing, it's an issue in these cases. And the last point is the cost of GMP phage products. The more phages are used, the higher the cost. And of course, and even so, not all phages used are effective on all isolates, because if you use a fixed cocktail, you will have in your cocktail phages that you already know that the phages are not active on the strain of the patient. So is it acceptable for us? Is it acceptable for the health authorities because of the cost? And is it acceptable in terms of selective pressure? for the selective pressure for resistant to phages. It's questioned we could have if we don't have or don't use phage susceptibility testing. So the best way for that is to use a cocktail of 2 or 3 phages. We prepare this cocktail extemporaneously just before to use in the patient. And the selection of the 2 or 3 phages that we will use have to be based on the real activity on the isolates of Mr. Dupont or Mr. Durand or what else. So we need to select the personal phages to be used, and for this, we need to have a phage susceptibility testing. It's clear. The second thing, it's also clear for our health authorities in France to obtain the access, the authorization to use phages in the context of the access authorization to compassionate use. You know, you have here the criteria of inclusion. Sorry if it's French. You need to perform the phage susceptibility testing before to use the phage, and to be sure that the phage that you use for the treatment are active on the clinical isolates. So, in fact, phage susceptibility testing, the regulator, what is very important for the regulator is to know the clinical trials are the clinical trials. The question is, are therapeutic phages active in humans? And if failure of phage therapy, what is the reason of this failure? And the question is to know if it's an in vivo inefficiency of therapeutic phages or bacterial resistant to therapeutic phages. So phage susceptibility testing, again, is required before administration during clinical trials for most of the health authorities. So I hope that you are convinced that phage susceptibility testing is something very important for phage therapy and the development of phage therapy. The question now is what still limits the implementation of phage susceptibility testing in our labs. So the techniques used classically in our labs are efficiency of plating. So for that you use phage dilution, you dilute your phages in serial dilution, tenfold serial dilution in medium. You put this on a soft agar medium prepared with fresh bacteria to be tested inside the soft agar. You put the serial dilution on your plates, and the day after you are able to measure to do a titer calculation. And for example, you have here the reference strengths using your phages. You know already what is the activity of your phages on these strains, and you compare with the activity of the strains of your patient, and you will measure the titer test strength divided by the titer of the reference strength. And for example, here you could see that there is a dilution of 10. That means that the EOP will be 0.1. And we consider that the phage tested on the clinical isolate has a high efficiency if the EOP is superior to 0.1. Intermediate efficacy if it's between 0.001 and 0.1, and no efficacy if it's under 0.1. But it's not so simple to do that, because you need to have manual techniques. It's a manual techniques. You have to prefer soft agar. It's not common in a microbiology lab. You need to have a delay of 2 or 3 days. You need to have a trained staff for preparation, for experiments, for PFU reading. It depends on the capacity of the reading, because sometimes the lysis plate is very small. And you need to also, something that's very important is to avoid contamination in the lab, because if you put your phage everywhere in your lab, for example, we work on phages, and 2 meters after, we have the National Reference Center for Staphylococci in my lab. So if one day the phage escape, I will be killed by my colleagues of the National Reference Center. So I have a dangerous job, in fact. The second technique is use of kinetic for bacterial growth inhibition in liquid media. So you put your phage at different concentration, you put your isolates at a fixed inoculum here, and you wait 1 day, and you will measure the inhibition of the inhibition or the growth of the bacteria. For example, you see here for this strain, you have without phages, you have an increase of the optical density. It's a simple measure of the optical density. For the different phages, for example, for the phages in green, you have a complete activity of the phages, no growth of the bacteria. For the red, you have growth, and after the strain is killed, and you have a decrease. And for the yellow, you have nothing at the beginning, but you have a small increase at the end of the incubation time. So if you stop after, and you have to compare the area under the curve, so each curve, and you compare to the no-phage curve. So the maximum is 100%. And we consider that the liquid SS score under 75% correspond to a phage active to treat a patient. The problem here is there is how you standardize the process. And if you incubate a little bit more, it's true result for one strain with phages, the 4 phages that we have selected for Staph aureus. You could see here that in fact for yellow, you could see here, you have a regrowth, a very late regrowth. For example, for red, you have an increase, but after a decrease, a disappearance of the bacteria. And if you measure under the curves, if you stop here, for example, this curve shows that it's not very good phages, but in fact, if you incubate more, the phage is able to kill the bacteria. And at the reverse, for yellow, you have the feeling, if you measure the under the curve, that this phage is very active, but in fact, a very late regrowth you obtain after 24 hours. So the question is with these techniques, again, you have a manual techniques, it's not an automate. You need to have 2 or 3 days. You have to have a dedicated kinetic or optical density reader. You need to have a trained staff for all the manipulation, and you have to, again, to avoid contamination. So it's not so simple. The question, for example, is to know which media to be used, which multiplicity of infection you have to choose to test. What is the cutoff to say that you have, for example, for LIS, we define that it's 75%, but it's very arbitrary at the moment. The question is there is no definitive correlation established between EOP or the liquid SS score with the clinical success or failure in humans. And it's very difficult to define interpretation criteria if we have not this kind of data. So it's the reason why, so what we need, what is very important in the future is what kind of data, what kind of experiments or studies we could do. We could do a multi-center performance studies for the repeatability and reproducibility of the techniques to see what is the how to say that? The solidity, in French it's solidity, the robustness of the techniques. We could do technical correlation between different techniques. We could do in vivo, in vitro correlation in preclinical animal study models, but what we are waiting is the result from clinical data, and to be able to correlate the clinical activity and the result from the phage susceptibility testing. And is the reason why it's very important. You know probably that 3 consortia have been funded last week by EU to perform 3 different clinical studies with 15 million for each study, 1 for bone and joint infection and 2 for UTI. And we are waiting this kind of results. And so the limit of the implementation is that we have no complete automation. It's really an issue because it's not possible in a classical lab to perform phage susceptibility testing. It's not without complete automation. You have limited skills and training required for the technician. It's the phage ready to be used. We need to have phage ready to be used in devices with a CE-EVD marked, with a long-term stability of the phages in the device, and we have to avoid contamination. So we have use a system, a device, very clean and very simple to use without risk of contamination. Because I remember you that with antibiotics, when you contaminate, for example, in liquid media, if you have contamination from one well, 96 wells, if you have a contamination from one well to another one, you have just a limited number, concentration of phages, so there is no problem in the lecture. With phages, if you have a very small contamination, as the phages is able to multiply, you have an activity in the vials contaminated. So the second thing, it's the overcost, because sometimes you need to have a specific, even if you have an automate, you will have an overcost of this automate, and for, in fact, a limited number of patients, so it's an issue in a microbiology lab in a hospital. And the last thing, it's in France, for example, PST is under medical biology regulation, and this kind of PST must be performed by a medical biology laboratory validated by a qualified, and the result have to be validated by a qualified pharmacist or medical biologist. So it's not so simple to go forward in this context. And it's the reason why ECAS create a phage susceptibility testing subcommittee to try to develop reference methods, quality control, and criteria for the interpretation of the results. So we, and I am very proud to chair this group. We have a steering committee of 7 people. Here we have 24 members with a gender balance and culture balance with many different countries, and we discuss about what could be the future. And we are setting study, for example, for the repeatability or reproducibility in different labs with different technicians. And we start just now a study with exactly the same media, the same protocols, the same phages and the same strains in 6 different labs. Yes, I will finish. The CAS committee will develop standardized reference methods, will develop criteria for interpretation of phage susceptibility testing, quality insurance, education and training. And it's clearly crucial for the development of phage-based therapy. I will not have enough time to do that. Anyway, I think that one thing very important. Many people speak about rapid testing. The question is it really the best way at this step? What we need is to have a good techniques for phage susceptibility testing. For example, if we have a very rapid techniques, if we stop here, we will miss this increase of growth. We don't know exactly what is the meaning in terms of clinical evolution of the patient. Here we will say that this phage is not active if we stop very rapidly. The method is very rapid, but in fact this phage is maybe active for our patient. Many people say we have to test biofilm, but in fact, is it really at this step very important? And is it also very important to test phage plus antibiotics? I have to remember you that even for antibiotics, we have not really rapid testing, no test for biofilm, and no test for association. So we have to think about that. I have not enough time to develop this kind of techniques. There is a technique using, for example, here, ATP plus luciferase with luminescence to detect very rapidly the growth or the no growth in presence of phages. Is developed by phage experts and by IntelliPhage. My colleagues in Switzerland develop a very specific method using nanomotion. You put the bacteria on a very specific system, and if the bacteria is live, the system has a vibration, and if the bacteria is killed, there is no vibration. So it's a way to measure, but it's a very complex system. At the moment, it's not possible to have this kind of machine in each country, or each hospital, but maybe it's the future. And last point, it's too complex. I don't have enough time, but it's a way to capture the bacteria and to be able to measure with light the fact the bacteria is killed or not in presence of phages. And with bioMérieux, we are working in a very specific techniques. It's a metabolovolatolomic component. It's a way to detect it's a nose. is able to detect if the bacteria we have a growth of bacteria or not, in presence or not with phages. And you could see here, you have here phages, no phages, a little bit phages, more phages, again and again, and it's pretty nice to see that the more phages you have, the less detection you have. It's 10 minutes. And there is a connection between this and this, but in fact, you could see here with optical density, we have a limited signal, but when the sensor at change of colors, there are no reverse evolution. It's not like for optical density. And to finish, so we have to standardize and fix the methods for the future. We have to fix the process. If we have to work with colony or with liquid fresh culture, for example, which media, which multiplicity of infection, which time of incubation, which reading, which criteria of interpretation, and we have to correlate with clinical data. We have also to fix quality control. That means we have to fix strains and phages to be able to do quality control in our lab if we want to do phage susceptibility testing, and we have to develop simple automated methods to perform that everywhere in our lab, and as soon as possible to help phage therapy development. Sorry, I was too long. Thank you so much.
So we've clearly run over time. I blame the chair entirely. So I'm going to be quick. Okay. Lego. Phages out of Lego. Okay. 3 days in jail. What does that say about your reputation? Or a rare stamp worth your... Correct. You guys are good. Okay, congratulations. Well done. Apology. So yeah, we have gone over time, unfortunately. We're going to have a panel discussion. That's obviously not going to happen.
I have to add that I spent 2 days in jail.
Oh, you want to clarify?
To screen for COVID.
So you say. Yeah. We thought we could just continue this. If you have any questions, to come and speak to the speakers afterwards. But we do have time for just like a couple. There's obviously the pitches at 6 o'clock. So we've got about 23 minutes. So if anyone does have a question for one of the panelists, would you like to? Here's your chance now. Wow, we were crystal clear. Oh, gone. Yeah.
No, at the moment we have no data between to correlate in vitro data and in vivo data, because in fact there is not so many clinical trials, and is the reason why we are very focused on the three projects that have been selected by AU for the future. But the problem, we will have the results in 2 or 3 years. So the best way should be to retrospectively to have an access to some strains used in patients to see to test with the reference methods that we try to fix, and to be able to know if what kind of criteria we have to use to decide if the phage is active or non-active. But it's not so simple, in fact, and we have not so many patients. It's like something probably in the world we have 300 or 400 people. And the phages are from different companies, and to be able to do that, you need to obtain the phages from the company, so it's not so simple. You need to have MTA and so on. You need to have obtained the strains, and the strains are no more available, so it's not so simple, but I think we need to have to do that.
That's perfect. I think, yeah, we should release you back into the world. Oh.
I was very...
Frédéric, okay.
Oh, sorry. Yeah, Frédéric Livet. Or Vieux. Sorry, yourself, anyway. I was quite surprised and relieved to see that some of the veterinarian use of phages, the experience is being translated now to human use of phages. There was a previous paper, well, it was a guideline criticising the lack of data from clinical trials, and said there were too many case studies and compassionate use sort of leading us on to use the phages. But really, that surely was, the problem with that surely was that very expensive to get through to clinical trials, so there will be a very limited number of those giving you the data. And so until we have the mechanisms to rapidly replace phages as resistance occurs, those clinical trials with fixed phage formulations will be few and far between, because you will see an element of failure just because you cannot adapt your product. So do you think now, with that adaptive, we will see more clinical trials? And will we then be restricted, unfortunately, by the same push-pull mechanisms? That's maybe another question. Push-pull mechanisms we see with antibiotics.
So just to confirm your question is, will with this new framework where we will be able to adapt the phages to the patient, it will work better? Is that you are saying? Because even in the clinical trials, there are some clinical trials in the past where there was this phage susceptibility test and the appropriate phage was provided to the patient. So what we can see on the landscape of clinical trials, there are some people who work on the fixed cocktail, but some other, few and a lot of, 50-50, I would say at least, who was working with personalized phage therapy, even within clinical trials. So your question is if it will be easier in the future? Sorry.
Yeah, do you think now, if that is adopted and you can change your phages, do you think we'll see a lot more clinical trials coming through?
I think today. Yes, but okay, you can. Yeah, yes, of course. So just different perspective. Today, when I see the hurdles to lead clinical trials is funding. And for me, regarding regulatory, when we see the openness, it's always case by case, so we can go to assessors, and we saw a lot of openness, even in the FDA. In the FDA, we know that even without regulatory framework, they allowed some new phages during clinical trials, okay? So just, I don't know if I answer to your question, but today what I see is more a question of funding problems than regulatory, my perspective. But Edaline, please.
So I think we are kind of in a dead circle, and why we have so few clinical trials, it's just like handful, like increasing, but still handful, is because a lot of the phages are really specific. So you would, and you cannot, make, as we saw, you cannot make a cocktail including like 100 phages and giving them. And so under the current rules, it's not really meaningful, like it's not doable to get a marketing authorization for each individual like phage. If you have an E. coli infection, for example, urinary infection, you will need to authorize maybe 50 active substances, differently. So it's not possible for the companies. They will be bankrupt even before they start, right? And the reason, so there is no meaningful way to get the authorization. And when there is no meaningful way, like to get authorization, then of course companies are not interested in doing clinical trials. But this will change, I hope. The European Commission recently gave 3 times 15 million euros for 3 projects. One of the projects that got funding is actually Estonian researchers. I'm from Estonia, so and they have a really nice design for the personalized treatment. So they have the E. coli urinary infection patients are the target population, and they have 50 phages, GMP compliant phages. And the design is really simple. So they do the susceptibility testing. Officially, the company within this trial will do the susceptibility testing at the company site. But they also actually try the susceptibility testing at the hospital site, just to see whether they are also they can make it work in the hospital. And so they select phages for each patient individually in this one arm, and they compare the patients who received the phages plus standard of care with the population that got just standard of care, and they are not comparing like each phage separately. They take the whole collection. So does the phage group work better or the same as the standard of care group? So, and I think this is a really nice design, and I hope that when the legislation changes, that opens much more avenues, and we see much more clinical trials for personalized therapies. Thanks.
Right, I'm going to draw it to a close, unfortunately. I'm really sorry it ran over. I hate it when that happens. I do apologise. Please do come and speak to us if you have any other questions. We're always happy to talk. And thanks so much to the speakers for preparing their slides and coming and presenting. Thank you very much. Have fun.